Hi, good evening, everybody. I'm Bobby, back with you. Yesterday, we were dealing with cellular metabolism under the topic of fat lipid metabolism and about oxidation of fatty acids. In fact, to put it more concisely, we were dealing with the topic of oxidation of triacylglycerol, and then after we had beta oxidation of fatty acids, right? And then about ketone bodies. So today we'll be slowly moving on to the table of synthesis of fatty acids, which is also complete. We're going on to the table of fatty acid synthesis and beta oxidation, right? After that would be protein metabolism, right? And the abnormalities in lipid metabolism. So topics dealt with today. And then would be biosynthesis of amino acids, protein catabolism, and under the topic of protein metabolism would be the transformation or transfer amino groups, and slowly would be concluding on this subject of cellular metabolism. After that, we would be going to cell reproduction, right? So before starting this, we'll have a small Q&A session like yesterday on what was taught yesterday, right? We'll answer those questions, get the answers, and then go into the subject matter proper and finish it. So just hold on for a moment. I'll be back with you, right?
Right, now we're moving to the subject of cell reproduction, right? Now let's give a briefing or maybe a little bit of a synopsis. Now all cells arise from pre-existing cells by a process of cell division. And there is a definite balance or ratio between cell that's a nucleus and cytoplasmic mass of cell called karyoplasmic ratio or nucleocytoplasmic ratio or cone-plasmic ratio. Due to continuous cell growth, this balance or correlation, a ratio is disturbed, which leads to cell division. Further, cell division is also necessary for continuity of organisms or species. The phenomenon of production of daughter cells from parent cell is known as cell division. A cell born after a division proceeds to grow by synthesizing new macromolecules and then reaches a stage where it is ready for division again. Now, before I can proceed any further, let me just add a little bit of Q&A to yesterday's portions. Just hold on, please, right? All right, for now, for some Q&A on inborn errors of protein metabolism, right? Now, categories of inborn errors of metabolism or internal errors of metabolism, IEMs of protein metabolism, are as follows. Disorders that result in toxic accumulation of any error of metabolism. Disorders of protein metabolism, example will be amino acid pathies, organic acid disorders, urea cycle defects, disorders of carbohydrate intolerance, and lysosomal storage disorders. These are all the categories of inborn errors of metabolism. Okay, others would be in specific. Now, what are the defects of protein metabolism? Right? There would be phenylketonuria, that's PKU. You can find out more. And then you have urea cycle defects, leucinosis, MSUD, methylmalonic aciduria or propionic aciduria, MMA, Barpia glutaric aciduria type 1, BGA, isovaleric aciduria IBA, homocystinuria HOM, type 1 tyrosinemia. And what is the error of protein metabolism? Inborn errors of metabolism are rare genetic inherited disorders in which body cannot properly turn food into energy. Then disorders are usually caused by defects in specific protein enzymes that help break down metabolized parts of food. Now, which of the following diseases are caused by an error in metabolism? Examples of inherited metabolic disorders include familial hypercholesterolemia, Gaucher's disease, Hunter syndrome, Krabbe disease, maple syrup urine disease, metachromatic leukodystrophy, mitochondrial encephalopathy, lactic acidosis, stroke line episodes, MELAS, Niemann-Pick. To have more on that topic, we could just check here. Well, we have an overview on that. We have just a little bit of those on the various symptoms. We've just mentioned these. Mitochondrial encephalopathy, that's MELAS, we just mentioned. Niemann, phenylketonuria. Next comes porphyria, Tay-Sachs disease, and Wilson's disease, right? Just a general mention, that's all, right? And then after that, quote those questions, right? And now, what is the most common inborn error of metabolism? PKU. Phenylketonuria is one of the most common inherited inborn errors of metabolism. Galactosemia, tyrosinemia, severe lysosomal acid lipase deficiency, and acrodermatitis enteropathica are just some of the few inborn errors of metabolism, right? And let's check for any other additions that we could have. There will be, those are commonly present with diarrheal symptoms. These disorders have multi-system involvement, so diarrhea will be one of many abnormal findings. Seek inborn errors of metabolism in a little bit of detail from the details from the US. An uncommon but noticeable exception is the child presenting with adrenal insufficiency. Neonates commonly present in shock with hyponatremia, hypokalemia, and hypoglycemia. And related terms will be, let's not go into that much of detail now. Let's revert. We've reverted. We're back to the subject matter proper, cell reproduction, right? Now, further cell division is also necessary for continuity of organisms or species. The phenomenon of production of daughter cell from parent cell is known as cell division. A cell born after division proceeds to grow by synthesizing new macromolecules and then reaches a stage where it is ready to divide again. Cell reproduction is more complex in eukaryotes than in other organisms. Right? The process of cell reproduction has three major parts. The first part of cell reproduction involves replication of the parental cell's DNA. The second major issue is the separation of the duplicate DNA into equally sized groups of chromosomes, right? And the third major aspect of cell reproduction is the physical division of entire cells, usually called cytokinesis. The division and replication of cells are the basis for both sexual and asexual reproduction in most organisms. All those changes which occurs during cell growth and cell division are collectively called cell cycle. The interval between two division phases is called interphase. The mechanism of cell reproduction or cell division is fundamentally similar in all organisms, showing kinship and unity of life. Now, in continuation, we have the different factors controlling cell division are cell size, karyoplasmic ratio, and mitogens, agents, factors, or substances that trigger cell division. Cell cycle, the sequence of events that involving growth and division. A cell undergoes from the time of its own formation by division of the parent cell to its own division into daughter cells is called cell cycle. Cell cycle is divided into two phases, the I phase or the interphase and M phase or cell division phase. Interphase is also called the intermitosis. is the longest phase in the mitotic cycle. Right? Interphase is the metabolically active stage and period of intense biosynthetic activity. Repair of damaged DNA sequence also takes place in interphase. Interphase is the most suitable period to carry out fish fluorescence in situ hybridization. Fish is used for detecting and locating gene mutations and chromosome abnormalities. The I phase is further divided into G1, first growth phase, S, DNA synthesis phase, and G2, second growth phase. In G1 phase synthesis, a protein, RNA, amino acids, ADP, and nucleotides occur. G1 phase is also called antiphase, as during this phase the cell stores ATP for cell division. The decision for cell division occurs in G1 phase. In S phase, synthesis of DNA and histone protein occurs. S phase is also called the invisible stage of M phase. In G2 phase, synthesis of RNA and protein continues and formation of macromolecules for spindle and organelle formation occurs. Now I'm just going to read out the table of the history of cell division, all the scientists who have made contributions. We first have Prevost and Dumas, 1824, who contributed to the first studied cell divisions. They described cleavage into fertilized egg or frog. Rimac, as 1841, found new cells developed from pre-existing cells while studying cleavage in eggs, discovered mitosis in 1855. Rudolf Virchow in 1855 to 1859 postulated cell lineage theory, that cells arise by division of pre-existing ones, that's omnicellular cellular. Strasburger, 1873, found out that new nuclei develop from pre-existing ones. Bavaria and Fleming, 1879 to 1880, studied details of somatic cell division. Fleming, 1882, coined the term mitosis. Brouwer, Sutton, and Van Benden studied the process of cell division before the formation of gametes, meiosis, along with Strasburg and Winnewater, 1887 to 1900. Farmer and Moore, 1905, coined the term meiosis. Grigoryev differentiated meiosis 1 and 2. Howard and Pelley, 1953, described cell cycle. Montgomery introduced the term synapsis. Montos, Gemosis, 1955, synapto-enamel complex. Right? In G2 phase, the quantity of DNA within the cell has increased to 4C, but the cell is still considered diploid. Metabolic activities in regard to growth of cytoplasm and constituents are occurring during G2 phase. During cell division, oxidative processes are minimum in M phase, and therefore deficiency of oxygen has no visible effect on the speed of mitosis. The GO phase is a period in the cell cycle where G cells exist in quiescent state. The cells which are not to divide further do not proceed beyond the G1 phase and start undergoing differentiation into specific types of cells. Such cells are said to be in G0 phase to distinguish them from G1 cells, which would soon enter S phase. Some of them, such as fibroblasts, which help in healing of wounds, grow and divide again on demand of the body. Some types of cells do not divide after attaining full division, differentiation, and finally die. These include nerve cells, skeletal muscle cells, and red blood cells. Cells that are permanent in the G0 phase are called postmitotic cells. An example of such cells are neurons. G0 state is the condition of a cell where division has been arrested at G1 state. Cells that have permanently stopped dividing due to age or cumulative DNA damage are said to be senescent. Cell cycle is regulated by cyclin-dependent protein kinases. Cyclins are proteins which activate protein kinases, regulating eukaryotic cell cycle. These kinases are synthesized in G2 phase and cause phosphorylation. of proteins of a nuclear lamina leading to breakdown of a nuclear membrane in the late prophase. P53 is a protein that functions to block cell cycle if the DNA is damaged. If the damage is severe, this protein can cause apoptosis, cell death. P53 levels, P53 levels are increased in damaged cells. This allows time to repair DNA by blocking the cell cycle. A P53 mutation is the most frequent mutation leading to cancer. An extreme case of this is Lee-Fromeni syndrome, where a genetic defect in P53 leads to a high frequency of cancer in affected individuals. P27 is a protein that binds to cyclin and CDK, blocking entry into S phase. There are a number of cell cyclic checkpoints beyond which the cell cycle does not proceed. A check on completion of S phase. The cell seems to monitor the presence of Okazaki fragments on the lagging strand during DNA replication. The cell is not permitted to proceed in the cell cycle until these have disappeared. DNA damage checkpoint sends DNA damage before the cell enters S phase, a G1 checkpoint. This is a flowchart on the cell cycle, right? Right, we have the interface there. We have the interface here with the non-dividing phase, right, also called intermitosis. And we have a period between two mitotic cycles, or preparatory phase or energy phase. You don't see, okay, let me just magnify it a bit more. We have the M phase, actual dividing phase, which involves two steps, the karyokinesis and cytokinesis. Then the plant cell, animal cells that undergo cleavage, which is the dead end there. Here under the interface, we have the G phase, S phase, and the G phase, G2 phase, G1 phase, S phase, right? And then we have the end of this phase called reaches antiphase, right? And then you have the mammalian cartilage, right? Somatic cells and the germ cells, which go in for meiosis. And here it goes, somatic cells go to mitosis. And here the mammalian cell, cartilage, diseased cells, etc., paramecium, etc., they go in meiosis. Right, so this is a cell cycle. Work and study on that in detail. We need that, right? And continuing the subject matter proper, here during the S phase and after DNA replication, a G1 checkpoint is also seen. Spindle checkpoints. Some of these that have been discovered are detect any failure of spindle fibers to attach to kinetochores and arrest the cell in metaphase, M checkpoint. Detect improper alignment of the spindle itself and block cytokinesis. Trigger apoptosis if the damage is irreparable. The spindle checkpoint blocks entry into anaphase until all chromosomes are properly attached to the mitotic spindle. The spindle checkpoint is an active signal produced by improperly attached kinetochores. The spindle checkpoint blocks anaphase entry by inhibiting the anaphase promoting complex. Cell division is absent in RBC and brain cells and highly specialized cells like muscle cells and nerve cells. In gymnosperm in development of gametophytes and embryos, nuclear division occurs, and after completion of nuclear division, wall formation occurs from periphery towards center. Actual cell division occurs in M phase, in which may be mitotic and meiotic. M phase itself compose a two tightly coupled process, mitosis, in which cell chromosomes are divided into two daughter cells, and cytokinesis, in which a cell cytoplasm physically divides. Karyokinesis, division of nucleus, is usually followed by cytokinesis, division of cytoplasm. The etaps of cell division. While they are cell division, cell reproduction, cell multiplication is the process of formation of new daughter cells from the pre-existing parent cell. Cell division occurs in three ways: amitosis, mitosis, and meiosis. In amitosis, division of nucleus occurs without visible chromosomes. Amitosis is an asexual means of reproduction in bacteria, banner fission, and in yeast cells budding. Amitosis is seen in the cartilage cells and some generating cells or diseased tissues, and in the old tissue, fetal membrane cells, etc. Drawbacks: amitosis, elongated chromatin fibers do not condense to form chromosomes. Elongated chromatin fibers break during constriction of the nucleus. There is no mechanism for separation and equitable distribution of daughter chromosomes. Mitosis causes unequal distribution of chromatin material amongst the daughter nuclei. It results in structural and functional abnormalities in the cells. Mitosis, the cell division that takes place in somatic cells, is mitosis. The site of mitotic cell division in plants are meristematic regions like stem tip, root tip, intercalary meristem, lateral meristem, growth of embryonic leaves, flowers, fruits, seeds, etc. In animals, mitosis is found in embryo development and some restricted regions in the mature form like skin and bone marrow. Mitosis is the process by which a cell separates its duplicated genome into two identical halves. It is generally followed immediately by cytokinesis, which divides the cytoplasm cell membrane. This results in two identical daughter cells with a roughly equal distribution of organelles and other cellular components. Mitosis and cytokinesis is defined as the mitotic M phase of the cell cycle, the division of the mother cell into two daughter cells, each the genetic equivalent of the parent cell. Because of cytokinesis usually occurs in conjunction with mitosis, it is often used interchangeably with mitotic phase with the word mitosis. Mitosis is called equational division because it results in the formation of two identical daughter cells, right? Because of the formation of spindle and visible changes in chromosomes. Mitosis is divided mainly into two phases: interphase and division phase. Phases of mitosis. For convenience of study, cell division is divided into four phases: prophase, metaphase, anaphase, and telophase. The prophase is early. Prophase is also called sparring phase. In this stage, chromosomes are overlapping with one another. In mid-prophase, the chromosomes appear coiled and shortened. The chromosomes appear to consist of two longitudinal fibers called chromatids attached at the centromere. The nuclear membrane dissolves and the nucleolus degenerates at the center end of prophase. In animal cells, two centrioles are formed from the centrosome. Spindle formation begins at late prophase. Metaphase is a short and simple. Centromeres are arranged in a row in the middle of the cell during pro-metaphase and mitosis, but the arms of chromosomes show deviation. During metaphase, fibers of spindle apparatus made of microtubules are three types: kinetochore microtubules, polar microtubules, and aster microtubules. The structure formed by the arrangement of chromosomes at the center of a cell during metaphase is called metaphase plate or equatorial plate. Spindle apparatus is formed from the microtubules of cytoplasm. In animal cells, spindle fibers are formed due to a division of centrioles where asters are present, so spindle of animal cell is called amphiaster. Spindle of plant centromere is called anastral. Spindle fibers extending from one pole to the other are called continuous spindle fibers. Fibers extending from a pole to the centromere of a chromosome are called chromosomal spindle fibers or discontinuous spindle fibers. Fibers which develop between the separating centromeres are called interzonal fibers. During anaphase, interzonal fibers are also called intrachromosomal fibers. At the daughter chromosome move towards opposite poles during repulsion between centromere, contraction of spindle fibers, and relaxation of interzonal fibers. Metaphase is the best stage in mitosis for analysis the chromosomes and to study their morphology. Graphic representation of a chromosome complement of a species is called karyogram. And karyotype anaplasmic helps in prenatal diagnosis chromosomal numerical abnormalities. That's it for today. Called the karyogram. Karyotypic analysis helps in prenatal diagnosis of chromosomal numerical abnormalities, gene location, or relationship between chromosomal change and developmental defects. Karyotype is prepared at metaphase of diploid somatic cells. Anaphase begins with the separation of the centromeres and the pulling of the chromosomes to opposite poles of the spindle. In anaphase movement, the chromosomes appear as V, L, J, and I shaped depending upon the position of centromere, and named as metacentric centromere is in the middle. Submetacentric centromere is partly middle. Acrocentric centromere is present at one of the chromosomes, and telocentric centromere is at the apex of the chromosomes. Division of centromere indicates the separation of chromatids. At the end of the anaphase, two groups of chromosomes are formed one at each pole. Chromosomes are shortest at metaphase while thickest at anaphase. The number of chromosomes at each pole is equal to the number of chromosomes in the paranucleus. Telophase is a reversal of prophase and prometaphase events. At telophase, chromosomes become condensed and uncalled and form chromatin. Nucleolus, nuclear envelope, and nucleoplasm is formed, and hence two daughter nuclei are formed at two poles during telophase. Sachs reported that temperature influences the process of karyokinesis in mitosis. Cytokinesis is the division of cytoplasm. Cleavage of foreign cytokinesis occurs in animal cells, where an invagination occurs at the equator, which deepens and finally cleaves into two daughter cells and are separated. Plant cytokinesis takes place by two methods: cleavage, usually in lower plants, and cell plate. In plant cells, cell plate is formed from Golgi apparatus between two groups of chromosomes. The cell plate grows from the middle towards the periphery and finally joins the cell wall. The early cell palate is the middle lamella of the many-layered cell wall. And saccharomyces, small protuberances arise from cell and part of nucleus after mitosis or amitosis migrate into the outer growth to form new cells. Mitotic poisons are inhibitors of cell divisions. For example, acids and cyanides inhibit prophase. Colchicine, vinblastine, and granosan inhibit spindle formation at early metaphase by inhibiting assembly and polymerization of microtubule spindle fibers. Chalons check cell division in vivo and in vitro both. A ribonuclease suppresses prophase, and heat shocks prevent cell division. The significance of mitosis. Essential for growth and development of a multicellular organism, helps to maintain the stability of chromosomes number and hence provides genetic stability. Method of multiplications of unicellular organisms. Mechanism for repair of old and worn out cells. Restores the surface of volume ratio of cell. Restore the nucleoplasmic index. Produces multicellular condition and provides opportunity for differentiation. And repair of body takes place because of addition of cells by mitosis. Types of mitosis. When karyokinesis is not followed by cytokinesis, division is called for free nuclear division. Repeated free nuclear division results in the formation of a multinucleate cell or a sinusoidal cell in unicellular organisms such as amoeba and yeast. Mitotic events usually occur within the nuclear envelope, which remains intact, and the nucleus divides by furrowing along with the cytoplasm. Such mitosis is called intranuclear. In multicellular organisms, the nuclear envelope breaks down, and the mitosis is termed extranuclear. Replication of chromosomes without karyokinesis is called endomitosis or endoploidy. Endomitosis results in polyploidy when chromatids separate from chromosome. Meiosis forms the basis of sexual reproduction and can only occur in eukaryotes. In meiosis, the diploid cell's genome, which is composed of ordered structures of coiled DNA called chromosomes, is replicated once and separated twice, producing 4 sets of haploid cells, each containing half of the original cell's chromosomes. These resultant haploid cells will fertilize with the other haploid cells of the opposite gender to form a diploid cell again. In meiotic cell division, reduction chromosome number occurs, giving rise to 4 haploid cells. It was discovered, meiosis was discovered at Strasbourg, and is by Farmer and Moore. I use the term. And if meiosis does not take place in an organism, the chromosome number will be double in each generation. Meiosis maintains the chromosome number as constant. A cell which undergoes meiosis is called a meiocyte. right? In zygotic or initial meiosis, which occurs immediately fertilization in certain protozoans, groups, fungi, and some algae.